Ancient viral DNA drives early embryonic development, mouse study reveals key findings. A groundbreaking study reveals that remnants of ancient viral infections embedded in the mouse genome are essential for the earliest stages of embryonic development. This viral DNA, known as MERVL, works with a protein called DUX to activate genes that allow cells to become any type in the body. The research also sheds light on why this same mechanism can cause severe muscle-wasting diseases if left unchecked. The role of viral DNA in development for decades, scientists have puzzled over junk DNA, sections of the genome with no apparent function. Increasingly, these regions are proving to be far from useless. In mice, a stretch of DNA originating from ancient retroviral infections, MERVL, is now understood to be critical for early embryo development. When activated by the DUX transcription factor, MERVL turns on genes that give cells the ability to become any cell type, a property called totipotency. This is crucial for forming a complete organism from a single fertilized egg. However, this process must be carefully regulated. Prolonged activation of DUX leads to cell death, mirroring the pathology of facioscapulohumeral muscular dystrophy, FSHD, in humans, a debilitating muscle-wasting disease caused by a similar protein, DUX4, remaining active for too long. The study is important because it clarifies how these seemingly contradictory roles of viral DNA and DUX function. CRISPR reveals the mechanism researchers used CRISPR activation, CRISPR, a gene editing technique that boosts gene activity without altering the underlying DNA sequence, to dissect the relationship between DUX and MERVL. By selectively activating each factor in mouse embryonic stem cells, they discovered that while MERVL alone grants cells totipotency, it lacks key developmental traits. DUX alone, however, produces cells that closely resemble natural early embryonic cells, suggesting it drives the initial developmental cascade. Further investigation revealed that DUX triggers cell death by activating the NOXA gene, which produces a protein that kills cells. Removing NOXA significantly reduced DUX-induced damage. This discovery is significant. MERVL does not directly contribute to the toxicity observed in muscle-wasting diseases. Instead, NOXA is the primary culprit. Therapeutic implications, given that NOXA levels are elevated in FSHD. The study suggests that inhibiting this protein could prevent muscle cell death and potentially treat the disease. Senior author Michelle Perchard notes that FSHD is complex, with only a subset of cells activating DUX4, even though all cells carry the genetic changes. Understanding why this happens is crucial for future research. Open questions and human relevance notably, MERVL is absent from the human genome. However, scientists suspect that other stretches of ancient viral DNA in humans may serve similar functions during early development. Whether human embryos use the same mechanisms as mice remains unknown. Researchers now plan to investigate how MERVL controls nearby genes and when it is deactivated during mouse embryo development. Comparing mouse DUX and human DUX4 will also be valuable. Answering these questions could clarify species-specific differences in early developmental regulation. This research underscores that what was once considered junk DNA is, in fact, a vital component of embryonic development. Understanding these ancient viral remnants may unlock new treatments for genetic diseases and deepen our understanding of life's earliest stages.